Okay, cool. Well, thank you all for coming in. Like I said, this is our presentation. My name is Robbie O'Donnell. I'm the Watershed Program Manager with the Anacostia Riverkeeper. This is our kind of water quality presentation, a webinar, informational webinar to go along with a water quality report that we produced and published this week that basically goes along with water quality work we did in Anacostia tributaries, specifically Sligo Creek, the Northwest Branch, and Long Branch. So basically tributaries around Silver Spring, Colesville, north area of, or the south area of Montgomery County there. And we did it with Friends of Sligo Creek and Neighbors of Northwest Branch, who I'll introduce here in a second. But we're going to go through the webinar, talk about the results, talk about some of the things we noticed and found this summer. And then towards the end, we're just going to have a quick question and answer discussion, answer hopefully any questions you all may have about the results or what you've seen or anything like that. Like I said, if you have any questions, please just put them in the chat as we're going through, and we're trying to answer them as we go, just if they're topical questions about what we're talking about right now. But with that, I will defer over to my compatriot, Christine, who's also with the Anacostia Riverkeeper, and then we'll introduce our partners as well.
Hi, I'm Christine Burns. I'm doing tech support back here. I help run our water quality program at Anacostia Riverkeeper, amongst many things. You've probably all gotten 1,000 emails from me. And yeah, no, I'm excited to be here.
And then we have our two partners that we worked with on this grant this summer, Neighbors of Northwest Branch. And you want to introduce yourself?
Yeah, I'll turn my camera on. Yeah, I'm happy to be here. Ann Ambler, I'm the current president of Neighbors of the Northwest Branch. And I say current. The position is open.
Good to know. And then Pat, are you here?
Pat?
I am. Hey, Robbie. Hey, Christine. Sorry about all that.
Perfect.
Yeah, so I'm Pat Rakowski. I am currently the chair of the Water Quality Committee with Friends of Sligo Creek, and I've been in that position for about 7 years now, and I'm glad to be here. And I want to thank ARC and Chesapeake Bay Trust for putting this on. I'm very excited to be part of it. Thank you.
Awesome. Great, Pat. Well, to start, we're going to give a quick intro of kind of why we do this water quality monitoring. And then we're going to kind of pass it off to Ann at Neighbors Northwest Branch and Pat to talk about some specific things on Northwest Branch and Sligo Creek. And then we'll get into the actual results about what we saw this summer, general trends we saw, and then any questions and anything like that you all may have. But kind of the importance of how we see it in Anacostia Riverkeeper of water quality monitoring. There are 1,000 reasons to monitor water quality. Us being a nonprofit and being kind of trying to protect our waterways, but also serve as Anacostia Watershed residents and visitors, kind of eyes on the river. Publicly accessible and usable data is really important to us. So not just data that's collected for the sake of permits or anything like that, which is good, but that data is also useful to recreators. That data is useful to people who want to know what's going on in their waterways. That data is useful to everyone. And so we believe if it's publicly accessible and usable, it should be accessible to everyone, really. But this also does help enforce watershed management, things like TMDLs, NIPT permits, all that stuff. It can help inform policy decisions, whether swimming or anything like that is legal or illegal. That's mostly down in D.C., but that's a good example. And then just overall watershed health. So is our aquatic life doing well? Are our streams in peril from stormwater? Is some stream green in color or something like that? All these things we need to know to make sure that we have a healthy Anacostia watershed or healthy Northwest Branch watersheds or healthy Sligo Creek watersheds. You go to the next slide, Christine. But specifically in the Anacostia watershed and by extension, the Capital Beltway region, as you all know, I mean, the DMV is just ransacked with roads and development. The Anacostia watershed in and of itself is over 70% developed, considered developed. In Montgomery County specifically, so the Anacostia watershed, the whole watershed, which you can see in that figure, is 176 square miles. So relatively small. When you put that into perspective of, say, the whole Potomac River watershed, that's 14,000 square miles. And then you put that into the scale of the whole Chesapeake Bay watershed, that is 64,000 square miles, give or take. So relatively small comparatively, but with over 70% of that watershed being developed, we are highly urbanized, as all of you may know, living and driving around all of our urban areas in D.C., Maryland, and Virginia. In Montgomery County specifically, so Montgomery County covers around 507 square miles, and 62 square miles of Montgomery County is basically lies within that Anacostia watershed. So a decent portion of the watershed, more so than in DC, actually lies within Montgomery County, in Prince George's County. So a bulk of the Anacostia watershed does fall within Montgomery County. So what this basically just means, and what I'm hinting at, what you all probably know, is that the Anacostia, and by extension its subwatersheds and tributaries, is beset with potential pollution from point sources, non-point sources, stormwater, salt that we saw from our non-existent snowstorm today, all these types of things. So we got to be kind of always vigilant and always careful about these things. Not that other watersheds in more rural areas don't have similar issues they're dealing with, but we kind of always have to be prevalent and always vigilant in an urbanized watershed, because we want to enjoy our waterways just as much as anyone else. But the threats and the potential for threats of pollution are much more ubiquitous, so to speak, around our areas. Next slide. And primary contact in Montgomery County waters does happen daily. So like I said, we monitor these waters for ecological reasons, permitting reasons, to help y'all know what's going on in y'all's waterways, but also because this primary contact of touching the water does happen in DC, it happens in Prince George's County, and it happens in Montgomery County daily. And so if everyone has more informed data about the waters, they may be able to make a more informed decision about their contact with that water. At least that's what we like to think. And so with that, I think we go into Anne and the Neighbors in Northwest Branch, telling us a little bit about the Northwest Branch.
Yeah. Hi. Glad everybody could come, and thank you to ARC for doing all this. We have wondered about what was in the water for quite some time, so it's given us a wonderful opportunity to join in the monitoring last summer and the summer before, where we were down in Prince George's County. Christine asked me to characterize the stream, the Northwest Branch, and how residents use it. And then, of course, I toss in what we do, and a couple of pretty pictures. So, Christine, next slide, please. Okay, this follows on to Robbie's slide showing just where the Northwest Branch lies within the Anacostia River watershed. It's on my screen anyway, it looks kind of small, so kind of hard to see, but I added all these arrows to point to where the roads are that cross, and where Sligo Creek is and Northwest Branch. And yeah, that little pointer helps. Okay, next slide. So at its primary source, at the Sandy Spring, it is basically just a little stream that goes across a field, a farmed field, before it goes into the woods and goes by the Underground Railroad. And then, actually, I guess earlier than that, it is joined by Bachelor's Run, which has lately gotten more developed. It used to be quite rural. And it goes on down, it picks up water from Belpre Creek, which drains at least one country club, and I'm not sure about the second country club, whether that goes directly into the main stem or whether it goes into Belpre Creek. But anyhow, and then it goes on down to Burnt Mills, where, as you all have seen, that dam, which amazingly is hollow inside and was part of the waterworks there when it was drinking water. Next slide. And there we have a view just downstream of Colesville of the Northwest Branch. And if you look just to the left of that rock that's sort of in the middle and come forward a little bit, I think that's our monitoring site. I didn't take the picture to be our monitoring site. It was a picture I had taken before, but I think that might be the site right there over to the left and forward of that rock. Next slide. Okay, so following, no, back up. Yeah. Okay, so next we go over the rocks. We fall off of the Piedmont onto the coastal plain. And then it kind of flattens out and slows down. And in Prince George's County, that picture over upper right is a very pretty picture in Prince George's County. Sometimes it's within a wooded area, and sometimes it's right next to recreation, as the bottom picture shows. You've got a service road there off to the left, and recreational facilities. And then it joins the Northeast Branch and becomes the Anacostia River. And the last picture, bottom right, is people recreating on the river. Okay, now the next slide. So how do the residents enjoy the Northwest Branch? Well, of course, there's a lot of hiking, biking, and bouldering. I didn't have any pictures of the boulderers. There's horseback riding. There's tubing and swimming. Against all odds. Tubing and swimming, fishing, and lots of people, lots of families bring their kids down on the banks, and then the dogs run in the water and the kids do some wading and playing in the rocks, and of course dog walking. So next slide. So what does that give us in the way of challenges for the water quality of the Northwest Branch? Well, we got draining 3 golf courses with whatever they put on their golf courses. Then we've got the ICC, the water from Paint Branch. In order to try to save Paint Branch, the water that falls on the ICC and the Paint Branch watershed is pumped over to the Northwest Branch. So we get all of that. Then we have the sanitary sewer running down, and that sometimes leaks. We've had 2 major reconstruction type activities, at least 2 by WSSC, to try to fix leaks most recently. And earlier up at Kemp Mill, they rerouted the stream and did major reconstruction of the bank because Superstorm Sandy blew out the bank so that the sewer line risked, the trunk line risked being exposed. Then there are crossroads. Now we don't have a road running alongside the way Sligo Creek does, but we have major roads crossing. In addition to the ICC, we've got Randolph Road in Colesville and New Hampshire at Piney Branch. And that gives us vehicle pollutants and litter, and of course in the winter, salt. And of course it is highly developed in Prince George's County. But the wonderful thing is you can be up in the gorge or between Colesville and Randolph Road. And in the summertime, when the leaves are on the trees, you can imagine that you are out in the wilderness. But you do have the dogs, the horses, and the goose poo from Wheaton Regional Park. So there are lots of threats to the water quality. Next slide. So what do we do? Okay, well, we have been lucky to be able to partner with ARC, with Anacostia Riverkeeper, to do the water quality monitoring now 2 summers. And Glenn and his team have been doing the macroinvertebrate monitoring for years now on the Northwest Branch. And then of course we do all our cleanups, we plant trees periodically. We have educational programs, we advocate, and we remove invasive, non-native invasive plants. Next slide. And that's it. Finally, we enjoy the Northwest Branch in every season. And thank you very much.
Robbie, you're on mute.
I was just going to pass it off to Pat. I know you're calling in, Pat, but we do have your slides pulled up.
Okay.
If you want to talk a little bit about FOSC.
Sure, will do. Yeah, great. Thanks to Ann. I couldn't see any of that, but it sounded like she did a great job. And it also, I think what you'll see when you look on the maps later that I think, Robbie, you will be showing, Sligo Creek essentially parallels Northwest Branch. It's more urbanized than Northwest Branch is, and it's shorter, but very similar. Most of the same major roads are involved, so lots of the same types of challenges to water quality that they see there with Northwest Branch, we have with Sligo, just perhaps a little bit more intense, a little more impermeable surface, and a little more urbanization. Briefly stepping back, just Friends of Sligo Creek, stats at a glance, we're about 20 years old, just a little bit over than 20 years now, about 600 members, split up into 6 different committees that focus obviously on different parts of trying to take care of Sligo Creek and also help people nearby enjoy it. We do outings, or used to before COVID, a lot of outreach and education, which has been difficult to do. We try to do it via Zoom when we can, but that has slowed down considerably. But we have kept up certainly with our invasives work, sweeping the creek for trash twice a year, and also with water quality and advocacy. So we're very active. We seem to be growing, which is always nice, and becoming more active also with our work with both the county government and the Department of Environmental Protection as well. So next slide, please, Robbie. And a bit about the creek itself, just circling back, it is about 8 miles long, meets up eventually with Northwest Branch, and then they become running into the Anacostia eventually. So it's in tough shape. It typically gets a poor to fair rating, although that is improving recently. On the IBI index, again, heavily impervious, super urbanized. There are buffers along most of the creek, but they tend to be very thin. They could be 50 feet. Sometimes they're several hundred feet. Other than the Sligo Creek Golf Course and up by Wheaton Regional, the park, the buffer for the riparian effort area tends to be pretty thin. So a lot of runoff, especially in the summer with flashy storms, that sort of thing is something we see a tremendous amount of. And therefore we do a lot of stormwater work as well to try to improve that situation if we're able to. And we see much of the same geography that Northwest Branch does. Most of the fall zone for us is sort of from Colesville Road on to about the PG County line, and then things flatten out pretty much there as you approach down towards Chilham and Hyattsville. And next slide, please, Robbie. So monitoring we've been doing to try to get a handle on what's happening to the creek. We're doing a lot of work in outreach and education. We do a fair amount of advocacy, both with Silver Spring and Tacoma Park, and also at the county level. But we had a sense that we might want to know a little bit more about what's happening in the creek, both from a macro invertebrate standpoint, also from a chemical standpoint. So we started monitoring about 10 years ago now. Began with 2 sites, we're up to about 12 now, and have expanded to include a variety of pollutants over time. Some of the most interesting ones lately have been chlorides and salinity. Obviously, with road salting, that's become a big issue for us that we're really trying to push on, along with the Isaac Walton League of America. They have a winter salt watch kit that we've recently started to distribute to various members, and we're really excited about doing that partnership as well. I know that's something that Anacostia Riverkeeper has been looking at as well, all through the watershed. So it's certainly an issue that we're concerned with and trying to work with the county and the state to change the management practices when it comes to salting roads and that sort of thing. We do share our data with DEP and with parks. We work fairly closely with them. And one of our monthly sites actually is one of their best biological sites for doing electrofishing. So we're trying to work with them to see if there's something special about that site that makes it such particularly good habitat. Just beginning that effort, but we're looking forward to finding out more as the months go by. And I think that's it for that slide, Robbie. Please move on to the next one. And this is just briefly a look at our current monitoring sites. Also kind of a nice look at how the creek changes over the course of its run. It first emerges in a place called Blue Ridge Avenue, and this is just slightly south of Wheaton Regional Park. Heavily impacted there. Everything has been covered over, probably back in the 1960s, I believe. And then during its course, it runs down through some fairly nice park areas in Wheaton Lane and Dennis Avenue. These are heavily used, which is, again, part of the reason we wanted to choose these sites is to see what the water quality was, where people were using it most. We've also added some tributaries that were fairly large along the way, including Flora Lane is the one you see in the lower left corner there. And then some of our legacy outfalls, the ones we've been looking at the longest, were also suggested by Department of Environmental Protection. That would include Bennington Drive, which you see down in the lower right corner. That again is at one time a free-flowing stream that's been almost entirely enclosed into stormwater pipes. And next slide, please. Same with Wayne Avenue, which is a difficult one, which will come up later, I think, as we discuss the results from last summer. That has chronic issues with coliform and chlorine, occasionally sediment, and ammonia as well. And then continues on down, getting flatter as we get to Fleetwood Terrace and Brashears Run, which is a major tributary coming down from Tacoma Park's downtown. So we've been monitoring that for about 7 years. And then continues on down, some beautiful runs, cobbles and riffles down by Jackson Avenue. This is a site that Parks and DEP monitor each year and has some very encouraging signs from a vertebrate standpoint. So we're curious to see what happens there with time. And then continues on down. Central Avenue is a Long Branch trib. That is our biggest trib coming in. We start to monitor that as well. And then finally down to 13th Avenue, which is right near New Hampshire Ave and almost to the PG County line. So those are our 12 sites at this point. And they started to give us a very well-rounded picture of what the creek does over time and what the impacts have been. And it's been super hooking up with Anacostia Riverkeepers and with Neighbors Northwest Branch, particularly at Fleetwood Terrace and Wayne Avenue, which are two spots that we've worked with in the summers to monitor with them. And next slide, Robbie. And just echoing what Robbie has said and others as well, it's just what we're finding is it's really helpful to have as many eyes as possible and noses and ears on the creek. And so our Water Watchdogs program is one example of that, which has been quite helpful, especially in the last couple of years with residents in the area, just keeping an eye out, seeing what's happening. If they see sediment, something doesn't smell right, something's going on a construction site, this is an automatic routing through email and also through text that goes automatically to us and to DEP. And we found it to be very helpful in kind of shutting down a couple of construction sites that weren't to code and other discharges perhaps from WSSC. So it's been a good system and we're looking to expand it as we can. And I think that's it for now. Thank you very much, Robbie.
Okay, so the current program that we have is 5 sites, and Pat did a pretty good job of outlining why we've chosen those sites. And this program we monitor every other week from about mid-May through mid-September. And so this summer, between sort of the bacteria and the other water quality parameters that we collect, we got about 350 data points. Which is pretty good. And then obviously Friends of Sligo Creek and Neighbors of Northwest Branch are our partners. So here's where all of the sites are located. You can see they're kind of in the southeast portion of Montgomery County. And I think something that was interesting that we did this year, and Pat kind of alluded to it, is in previous years, we had monitored the Sligo Creek 1 and 2 sites, and we added in Sligo Creek 3 just upstream of Sligo Creek 1 because we had seen high bacteria at the Wayne Avenue Sligo Creek 1 site in the past. Rami, is this you?
Yeah, this was just basically how we came about. So we didn't just all of a sudden fall into
Sit here and listen
water
to
quality.
it's a water monitoring quality.
We didn't just fall into a water quality monitoring program. This program, which is very beneficial in that this data being produced, like many other programs, can be given to DEP, like Pat said, or DC government if we're in DC, or any other place, and be used actively in policy decisions and regulations. So in order to do that, we needed to design the project, have a government approved QUAP and stuff like that. Train our volunteers, get our monitoring plan set up, and then actively report out the data, not just to the government, but to you, the constituents, as well, because you're the ones who are really actively interacting with these streams and waterways every day. And so, like Pat was kind of saying, y'all are our eyes, you're our ears, and kind of there's a two-way relationship between the monitoring and the volunteers who actually help us go out and monitor or report pollution or anything like that. Next slide. It's me or you, Christine?
Could be you.
Like we said, what we collected for under this program was for what we'll call recreational water quality. So is the water safe for me to touch or interact with? I.e., I can splash it on my face, I can go swimming, I can go wading, etc. And so for recreational water quality, that's air temperature, water temperature, pH, bacteria. Specifically, we do E. coli and turbidity. Mostly what we focus on is pH, E. coli, and turbidity. Those are the three primary parameters that affect recreational water quality, i.e. if any of those were to be failing, it would not be safe for us as humans to go swim or anything like that. On top of that, we also collect ecological data, so stuff like dissolved oxygen, specific conductivity, getting back to that chloride dissolved solids parameter that Pat was back talking about, and things like salinity and all this other type of stuff. So this is going to tell us if the waters are safe for not just humans, but for our aquatic life, our macroinvertebrates, our small fish, things like this that are actually living in the water. So we kind of wanted to get a double approach to this to get a broader view of the streams and see if they're safe, not just for humans, but also for the life that's living in there. All right, next slide. And then finally, for our bacteria, which is our primary parameter, we do this in a very similar way to how you may have done something back in like a 10th grade biology class with the agar discs that you see with the little bacteria colonies growing on them, and it's the quintessential science picture that you may see in a science textbook. The process we use is very similar to that. It's more of a statistical method, but we essentially take a sample with a bottle. We put a special chemical in it that allows the bacteria to fluoresce under UV light, which you can see in that bottom right picture. This whole setup, I could train every single one of you how to do it in a very short amount of time. But this setup is not just used by nonprofits, it's used by local, state, and federal governments. It's a very common system in how people process E. coli and Enterococcus nowadays. So this is the IDEX method that we use. You may see it sometimes at your vet. The vets also use it. It's used in the agribusiness as well. So this is giving us our E. coli levels that we actually get. Next slide. And then finally, when we present the data, so we go out, we collect all this data, our volunteers go and collect the samples. I may be out there with our water quality probe testing the dissolved oxygen or the conductivity or anything like that. But when we're actually reporting out this data, so every summer you can look on our website, you can look at our social media. We report our data to an app and a website you can actually get on your phone called Swim Guide. We also report it to an app you can get on your phone as well called Water Reporter, which is also very good for reporting pollution. So these are all good, kind of nice technologically advanced ways that water science is growing, so to speak. But when we post our results, we say if things are passing or failing. So as an example, Sligo Creek 1 is the Fleetwood Ave or Fleetwood Terrace site that Pat was talking about. So that's on a nice little part of Sligo Creek. Unfortunately, it has quite bad bacteria. So when we report if that site was passing or failing, we'll give you all the data, but we typically like to report it in a pass-fail way. And typically we're reporting this based off of the bacteria. These limits you see in front of you are basically the primary contact thresholds. So these are values in green that tell you if the water is 100%, or maybe not 100%, but safe to swim in. And the values in red are basically essentially would be not safe for humans or even your dog to go swimming in. So basically any bacteria that's above 410, any pH that's outside of 6 or 8.5, and any turbidity that's really above 20 NTU or FSU or whatever turbidity unit you want to use. These are considered unsafe for primary contact. And we like to just boil them down into a nice red-green scenario. All right, perfect. So go ahead, Christine. We can get to the results now. I know everyone's been waiting with bated breath.
Yeah, drum roll. So we just put together a brief water quality report. That's a screenshot of the first page, and now that's live on our website. So that's our little PSA. And I'll drop it in the chat once the webinar is over, so you guys can download it if you want to. But the gist of it all for bacteria, at least, is on the right-hand panel of this. It's the location of each of our sites and then the percentage of times that they passed this summer or failed this summer. And so you can see that 3 of our sites, Northwest Branch, Sligo Creek 3, and Sligo Creek 2, passed about 50% of the time, which what I would say is kind of like meh. And then Sligo Creek 1 only passed 30% of the time. That's the one that was sort of had poor water quality in the past, and we were trying to tease out the relationship between Sligo Creek 1 and the Wayne Avenue outfall. And you can see that Sligo Creek 3, which is just upstream of that, does pass a little bit more often. So the outfall is definitely contributing, or likely contributing, to the high bacteria at Sligo Creek 1, but none of the sites are doing stellar. And then the Long Branch site did quite poorly this summer. It only passed 1 time. And that's a site that the Audubon Naturalist Society has been monitoring for macroinvertebrates for the past couple of years. And they sort of confirmed that their assessment of the water quality there was also not great. And so sort of doubly confirmed. These are the average bacteria levels through the summer, and then the red line across the screen is that 410 threshold. And you can see on average, you know, the water quality is pretty high at all, or the bacteria levels are pretty high at all 5 sites. And then with like Long Branch and Sligo Creek 1 being the worst of the 5. For bacteria in general, especially with regards to recreational water quality, there's a pretty close relationship between precipitation and bacteria. And so, you know, it is not surprising that we would see high bacteria levels after a rain event. And lots of places put out advisories that say, you know, in our area, you don't really want to have primary contact with the water within 72 hours of a major rain event. And so this graph is on the left-hand side, shows the bacteria levels, and all of the dotted lines are the each of the different sites throughout the whole summer. And then the right-hand side shows the precipitation, and the big blue bars are the actual precipitation levels. And you can see that on July 28th and August 11th, for example, we had 1.5 to 2 inches of rain in the 3 days before monitoring, and all 5 sites failed. So not surprising at all. I think what was, particularly of interest for us this summer was we had a couple of instances where there was very little to no precipitation in the 72 hours before we sent monitors out, and we still had you know, high bacteria levels at a number of sites, and those let you know that you know, there are lots of other contributing factors to the bacteria, not just the, you know, the stormwater outfalls.
And Christine, Kem just asked in the chat, what outfall were you just referred to? So the outfall is at the one that Pat was talking about at Wayne Avenue, where it crosses Sligo Creek there.
Just so between essentially SC1 and SC3.
Yeah. So SC1, when we were monitoring last year, we noticed that SC1 had very unusually high levels, or I guess maybe not unusually because they'd seen it before, but high levels of bacteria. And so this was just directly downstream of that Wayne Avenue outfall. And we wanted to see, okay, if we sampled above that Wayne Avenue outfall, would it be the same? Would it be different? Anything like that? And we did notice that on average and in terms of pass-fail, the results for SC3 were typically a little bit lower when compared to SC1. I can't, Pat, is that Wayne Avenue outfall a WSSC sewer? Is that a stormwater outfall?
It is a stormwater outfall. Mm-hmm.
Okay, correct. And so we wanted
That's
to see the relationship there. And it is proving, I think we want to sample it a little bit more, obviously. But there is something interesting, for lack of a better term, there. And we did, Pat, y'all sent that letter last year to WSSC, correct?
Correct.
And so a letter has been sent.
Yeah.
If you want to explain that, Pat, a little bit.
Yeah, it's Wayne Avenue. We've had very high readings there for coliform as well as many other pollutants over the years. But we seem to be stuck a bit in an administrative cycle where the WSSC says it is a stormwater pipe, but they don't have any jurisdiction. Essentially, once it hits the stream, it's not their issue anymore. It's going through parked land at the end. It goes underneath a middle school there and an elementary school on its way. It suggests, given the levels of bacteria we've seen, that there's probably cross connections in there. That's our guess, our best guess, that may be decades old. But the time and expense to go all the way through the system to try to find them is, I guess, something no one's eager to do. I think that's the quickest way to say it. So we do know that outfall has been a problem child for a long time. We're not sure why, and we're having trouble getting much in the way of movement on it, but we will continue to try.
Exactly. And that's, again, a benefit, a small benefit, but still a benefit of programs like this, is able to produce this data that every single week we send to folks at DEP and post publicly and send to folks like y'all and say, hey, if there is something potentially nefarious, not nefarious, but something potentially worrisome going on at this area of Sligo Creek. It allows us as partners to keep constant pressure on it and keep the issue in the public's eye, so to speak. And to give people... Yeah, exactly. I mean, everything looks so nice and picturesque, but in reality, there could be some dangerous bacteria there. And to give people kind of some more spatial orientation, we talked about the Northwest Branch site that Ann was talking about. Hold on, there was a question. Hey, Pat, Kem asked, has anyone considered inserting tracers into the school bathrooms to see if they're the ones as the illicit hookups?
Yeah, it's a great question. And yes, they have. And they did not find the illicit hookup there. I think what it is, too, is WSSC, they are water and sewer. They are not stormwater. So what we've heard, I believe, from them, and this is more our advocacy team takes this part of it, but my understanding is their position is this is a stormwater outfall. We don't do, so that's not us. We do sewer and drinking water, but stormwater is not our jurisdiction. So we're sort of lost in a netherworld there with that one. But the dye tracers we have tried, and we've tried that at another hotspot along Maple Avenue in Tacoma Park. And DEP, Alex Torello at DEP, I think went to 22 different apartment buildings and stores, tried dye tracing on all of them, and none of them came through. So they can be very difficult mysteries to solve.
Indeed, as Ken said, an interesting black hole. Very much indeed. But that, again, is the high benefit of programs like this, is keeping stuff in the public eye so these issues keep getting worked out. Yes. They don't disappear. Yes. On top of the... Go ahead, Pat.
No, I'm just agreeing with what you say. It's important just to keep it in the public eye. And we found some county officials are more open to it, and concerned than others. So that's helpful as well. So we get folks who can... hopefully get interested in it and keep after it.
Indeed. But in addition to the bacteria and the recreational data that we collected. We also collected ecological water data with our water quality probe, and that we actually found, in terms of the actual physical data, did have relatively normal to good ecological water quality data. So average dissolved oxygen across all sites was above 8 milligrams per liter, which is very good and ideal for aquatic life. Average specific conductivity, which really is the one you want to look at when it comes to chlorides, road salt, stuff like this, dissolved solids and everything, was perfectly normal for freshwater streams. Average pH was slightly basic, but almost exactly at a neutral 7, at average of 7.4. And average turbidity was very, very low at 3.1, so relatively clear streams. Obviously, these averages can be slightly skewed because maybe we didn't sample on a super wet day when there's maybe a lot of turbidity or something like that. And obviously, we're not sampling these streams during the winter time when the specific conductivity can be thrown out of whack by road salt. But that's what's important about other programs like the Isaac Walton League Salt Watch and stuff like that, is that those are able to cover more of those times to see those cycles and things like specific conductivity and everything like that. What's interesting about this, and what Christine alluded to, was all this ecological water quality data seems to point to good aquatic life. We got some nice photos of some nice aquatic life and terrestrial life wandering around our streams here. That was a snake we found at Northwest Branch. But at the Long Branch site, which is right next to the Rolling Terrace school there, it is just downstream of an outfall. It has bad to horrible bacteria levels. Although the ecological levels are normal and all within range, the Audubon Natural Society's microinvertebrate testing seems to indicate that the water quality is poor to fair, basically. It's not totally terrible, but it's not great in terms of the aquatic life that are actually living there at that site, which is an interesting juxtaposition. So that's another site that we want to try and keep in the public eye, and that is a smaller tributary that flows into Sligo Creek. So there is definitely some interesting stuff going along Long Branch there as well. Next slide, Christine. So finally, to sum it all up for everyone, across all the sites, we saw fair to poor recreational water quality. Troubling trends given the sites. All of these sites are in very close proximity to highly recreated areas, whether they're trails like on Sligo Creek, whether it's schools, whether it's parks, whether it's bouldering fields in the Northwest Branch, anything like this. All these sites were selected because they're so close to highly recreated areas. So the potential for contact with that water is very, very high. The Sligo Creek at Wayne Avenue situation still is troubling. We noticed that bacteria levels were up to 25% lower upstream of that outfall when compared to bacteria levels downstream of that outfall. So we definitely still want to continue investigating that. The Long Branch site, as I just said, was the worst when it came to bacteria, and also had that very interesting kind of ecological question as to why macroinvertebrates were saying that the water quality was poor to fair, but the ecological water quality that we collected seemed relatively good. So there's a little bit of a quagmire going on there. But finally, across most of the other sites, the ecological life did seem relatively healthy. YSI probe that we use picked up pretty normal results for a lot of the ecological water quality. So we'll continue to keep an eye on that as well. Mostly to continue to collect baseline data, but also to record any spikes that we may find, where if we notice that an average dissolved oxygen is 7 to 8 at a site, but all of a sudden we see a 5 or a 4, we know that's troubling. And then next slide. So finally, a lot of this data can be accessed in a bunch of different locations throughout the summer when we collect it, as well as right now. On the Chesapeake Monitoring Cooperative, which is a data explorer type site done by the Alliance for the Chesapeake Bay and the Chesapeake Bay Program. Water Reporter, which is an app done by a group called the Commons. Swim Guide, which is a very similar app, and on our website as well. Finally, what's needed, at least in my opinion, and y'all can chime in on what you think, is more detailed precipitation data. As Christine was talking about, that precipitation data and trying to find the difference between dry days and wet days and what's causing a lot of this bacteria to flow in is important. bacteria source tracking, or eDNA, determining if the bacteria is coming from human sources or avian sources or pet sources or equine sources. Things like this is really important. It's a hotbed of research right now in the Anacostia and other streams. And we're trying to figure out it in DC. We're trying to figure it out in Prince George's County. So it's very popular around the watershed, but also across all streams in the US and the world. Targeted signage around this area has been talked about. We maybe like to see it a little bit more, maybe see more advanced talks going into that. But something about, they have it in Rock Creek, something along the lines of there is potentially dangerous water here in times of high precipitation or things like this. And then finally, targeted investigations around areas with high bacteria. This goes back to the Wayne Avenue site and things like this, just to see if there's any sources, point source pollution we're missing, that they're missing, anything like this, whether there's any of these illicit hookups. Pat was saying these can be some tricky little investigations to do, year-long investigations to do, but projects like this help keep everything in the public eye, hopefully. So that progress will be ongoing, but continue to do these projects and hopefully pushing towards these targeted investigations, maybe a little bit more, can help elucidate some of those issues. So I think that should be it. So thank you to everyone. Thanks to Pat and Ann and Neighbors of Northwest Branch and Friends of Sligo Creek. And thank you to Chesapeake Bay Trust and Montgomery County who helped fund this. This is a great grant, and hopefully we'll be able to do it again next summer. In the chat, I believe Christine just put in the water quality report. And with that, we will open it up to any questions, comments, concerns, anything like that y'all have. I will look in the chat right now and see what we got.
Feel free to turn your mics off and stuff for questions.
Pat, this may be a good question for you. It says, what role, if any, does park planning have to do with water pollution? I live in the area covered in tonight's presentation. Our county council and county exec are up for re-election. How can we get some good talking points for candidate forums?
That's a good question. I believe, there's a lot of great material online, both in the broader context of urban planning, park design, that sort of thing, as well as water quality, water pollution. But I think also at the Anacostia Riverkeeper site, Neighbors of Northwest Branch, our site, there are a lot of groups in the area that you can find out a lot about health and the status of waterways in our area. I do think, I've come to believe over time, being heard is important. And I do think if you approach council members or even folks at DEP or parks with your concerns, it makes a difference. They know that people are watching and that it matters to them. So I would suggest that to you. Whatever issues you're concerned with, you should definitely write them, text them, give them a call and let them know. And I think that's how it starts, because we have found that if we're vocal and they know we're watching, it does make a difference.
Any other questions, comments, or anything like that?
Yeah, this is Janice. I have, that was a really good presentation. I really appreciate it. So I just remember the reason we did this is because we saw a lot of people swimming, during the pandemic because the pools were closed, et cetera. So I'm just trying to think about, what is a little confusing to me is that you said overall, the bacteria levels were pretty high. There was a lot of failure. And then you kind of summed it up, said fair to poor quality, but the ecological part you said was pretty good. So I'm kind of having a hard time understanding why, those certain invertebrate or those certain critters can feel like they can live there, but it's not good for humans. Could you explain that a little more?
And that's where, recreational water quality and ecological water quality can be a little different at times. If you imagine them as Venn diagrams, obviously they overlap a lot. The ecological water quality data, stuff like dissolved oxygen, specific conductivity, that isn't going to be, if a water has low dissolved oxygen, that's not going to be detrimental to human health, because that's not really a chemical or biological thing that's going to impact our biology if we're swimming in it, i.e. seawater has 35 parts per million salt, so it has relatively high, or parts per thousand, salt, and has relatively high specific conductivity. But when it comes to things like bacteria, things like E. coli, which is basically the bacteria that's coming from any warm-blooded animal, humans, dogs, whatever, that is going to be more detrimental in a biological sense to humans because that's impacting our biology more than it's going to impact, say, the biology of a tadpole or something like that. At least to my understanding.
Well, and the issue with the bacteria, right, is that if you're swimming in it and splashing in it, or your dog is playing in it and then you're petting your dog, like, you know, it's really easy for you to accidentally ingest a little bit of that. And, you know, then the bacteria gets into your system and you wind up with a gastrointestinal problem, right? And so that's where one of the big issues between the bacteria data and the ecological data. And also, I think I would add that the bacteria moves through fairly quickly. It's like a very spotty thing. So, you know, you might have an incident that brings in a high amount of bacteria one day, and then that's not there the other day. And it can be kind of, at time. I mean, obviously, if you have an extended sewer leak or something like that, you may consistently have high bacteria. But I think one of the challenges with a bacteria monitoring program and why it's worth it to monitor weekly or every other week is because it can change from day to day pretty quickly.
And it's a good example of like why they have things like Class A waters, Class B waters, Class C waters, or like all these different qualifications for waters. Because some streams may be perfectly healthy to swim in and be great for aquatic life and all this stuff. While some streams maybe you don't want to touch with a 10-foot pole in terms of going to swim in, but still have perfectly abundant and healthy aquatic life. And that's just how water quality works sometimes. So that's kind of the small select differences between ecological and recreational water quality and why one can be fairly good and the other one can be pretty bad.
Thank you. Yeah. So, I mean, if people are still swimming in it, what would you say to them? Like, it's not really good to swim in it? What?
Yeah, I mean, so that's almost comes to like a sociological thing. You know, we at Anacostia Riverkeeper, me, Christine, and our Riverkeeper Trey have had many conversations, whether it's with swimming and fishing, and no one ever likes to be like told not to do something or like that type of thing, or like almost come across as accusatory. But we just try to inform people. We say, hey, you know, like because these streams are in urban waterways, they can, you know, stormwater comes into them, which can carry dog waste or anything that's on the ground, stuff like this. We try not to maybe tell them like, hey, don't swim there. Like in DC, as it being illegal, we don't say, hey, it's illegal to swim there. We may say, hey, the water can be potentially dangerous to humans at time or things like this. Or just having a conversation with someone and educating them a little bit more than they may already know about it. Because sometimes you may not get a reaction then, but they may go back home and be like, oh, okay, I maybe need to think about this or do a little more investigation before I go wading or swimming in some of these waterways.
I would advise that, like, you know, if someone was like, you know, I really don't care, I really want to be wading or swimming in this river, then at the very least, making them aware of the 72-hour rule for our waterways is at least a plus. It's not going to catch every instance of high bacteria, but it will help with some of the most predictable ones. And then, like, if I had to give like a pro tip or something, I'd say just be really good about showering, hand washing, not touching your mouth, your face, etc. after having touched the water.
Okay, thanks.
Not that I think we should be giving out pro tips, but
This is Pat, and I would just echo what both Christine and Robbie have said. That is probably the most frequent question we get when we're out monitoring: is it safe to have my grandchild or my child go in here? Can my dog go in here? And we give essentially the same answer: the 72-hour rule with storms, and we can't recommend that you go in, but if you do go in, just make sure you wash well afterward. Don't let your kids put their hands to their mouths, that kind of thing. because people, it's so attractive, and that we don't have many streams in such an urbanized place, and so many of us just feel like, I got to go in there, and it's hard. And we saw the same thing last year with COVID. People were using it for swimming and bathing and fishing and everything that we'd never seen before. So I think just making them aware, and as Robbie has rightly said, no one really likes to be told no, but it is, I think, important to just keep people aware of some of the risks if you do it, and some of the tips, as Christine said, if you do go in, what to make sure not to do and what to do afterward. So I think that's important, and given that most people are going to continue to allow their kids to wade in or their dogs to go in.
Can I ask a question?
Of course.
Are you guys ever going to do what it takes to determine what kind of E. coli is there?
So the source tracking. So we want to try doing that. I know with relative certainty, I know it's occurring in D.C. I don't know about Prince George's County. If I had to guess, and again, I don't know with certainty, I believe Maryland or Montgomery County DEP is doing some type of eDNA tracking. We've done it before on the Anacostia main stem a few years ago when it was still kind of, the technology wasn't fully there, I guess. And we didn't find anything overly conclusive, but we're going to try to do it again in this upcoming monitoring season. But it is important into kind of how you're alluding there, Anne, and to do it not just in the main stem, but to do it in these tributaries where you can find, where that data would be almost put to more use, so to speak, a la if we found human bacteria at that Sligo Creek site or at Wayne Avenue at that outfall, then you could probably make a pretty safe assumption that it's some type of illicit hookup into that outfall or something like that. So the sources of the pollution are equal, but also maybe easier to find if they're on a tributary, where you know where the stormwater hookups are going to and you know exactly what's around it, as opposed to a main stem where it's collecting everything from upstream. So
Yeah.
hopefully it's a little more expensive.
Well, what I'm thinking is if for instance, the testing showed that there was a large amount of E. coli from dogs, it would bolster the argument for collecting your pet waste.
Right.
Because I know a lot of people go and walk their dogs along the Northwest Branch, and I don't think they always collect the pet waste. Figuring that, well, it's in the woods, so it's a natural phenomenon, and it doesn't matter. And then also, if we could find that there's an awful lot of goose poop from coming in from Wheaton Regional Park, and I guess maybe other places, but particularly the park, that would at least be useful information. I doubt that there's
really
very much. Yeah, go ahead.
It's really good for like those exact policy decisions. It's like it actively gives good evidence to go to policymakers, regulatory agencies, whoever, and say, hey, like in Northwest Branch or whatever, like we notice a distinct avian source coming into this waterway around this area. Like it gives you credible evidence and a credible lead to then go and enact some kind of investigation or change to.
So we are looking for partners on that, but we don't have like the lab facilities for that. So that's us needing to find somebody who has a much fancier lab than our own in order to be able to pursue those things.
Well, speaking of, this is Pat again. Speaking of that, I think you all know that WSSC for about 10 years did PCR testing, source testing on DNA for coliform all through the watershed, part of a consent decree for the MS4 permit. This is like in 2006, 2007, somewhere there. And they did testing at a variety of spots, I think for 8 to 10 years. And it was a mix of sources, as many people I think are alluding to. It was a lot of it was avian or canine. There were certain places it was maybe human was 20 or 30% of it, but often it was 5 or 10%. So it did, from what I recall, the data seemed to point more toward either avian, canine, sometimes ungulate, so deer, and then other.
was the vast majority of it. So I think, Robbie and Christine, you're exactly right, that it does help a lot with policy. It is expensive, or it used to be very expensive to do. I'm not sure at this point if it's something that's maybe a little more in reach. But and the policy part of it can be really useful to know those numbers.
Yeah. On the same subject, this is Paul Morawski. The comment in the text chat about purple line construction polluting Sligo Creek. I mean, how would purple line construction introduce bacteria into the mix? I mean, it seems to me that there's some other source that was just sort of conflated with purple line construction. Is that a crazy conclusion?
Robbie or Chris, who would like to take that one?
Look,
Pat,
go for it.
Yeah,
okay,
I'm not exactly sure about that.
Sure, I didn't want to jump. Just we monitor actually right next to some of the purple line construction. I know that sediment has been an issue, sediment runoff from improper fencing over the years from the purple line. So that may be what they were referring to. I'm not sure what the source was. As far as coliform, yes, I would agree with you. Unless they disturbed a pipe, accidentally struck one, or dislodged it, or changed the seal on a gasket or something. But yes, typically with sediment was what we saw from purple line. That was an issue. As to coliform, I agree with you. I don't think there would be a direct or an obvious connection there.
You're talking about turbidity.
Yeah, got it. Okay, thank you.
Welcome.
Anyone have any other questions or comments or anything like that?
Yeah, this is Janice. I just wanted to know, I think this would be really useful, like our civic association would be really interested. Do you come and, do this presentation to other organizations? Is that something you could do?
Yeah, we can. I mean, we at Anacostia Riverkeeper, and I'm sure Pat and Ann say the same thing, try and spread whatever water quality data, concrete water quality data we have to whoever will listen. We obviously target that a little bit at constituencies and DEPs or DOEs or DOEEs or whoever. But we will be more than happy to share this presentation, come give the talk, anything like that, for anyone who really wants to listen.
Okay, thank you. Yeah, I've been talking to the president of the Civic Association at Four Corners, and yeah, she didn't know what was going on and said she would be interested. So I just wanted to ask you all. Thank you.
Janice, reach out if you.
Yeah.
Are you in the North Four Corners Civic Association?
Yes.
Okay, because they just invited me to come and talk at the February meeting.
Oh, is that right? I had been talking to somebody several months ago. So, that's awesome. Well, okay. So you're going to talk about it then?
Well, it really should be Robbie who talks about that, but they've also invited Trey. But especially
Trey
to
goes
talk about the litter trap.
Oh, true. Yeah.
Oh, well, I'm glad to hear all of this.
We try and get out as much in the upper watershed as we can.
I just dropped our emails in the chat. So anyone who wants to contact us, it's just our first name at anacostiariverkeeper.org.
Oh yeah, I have another question about, about environmental justice. I mean, what do you have like any, connection to that in this, in what you do?
Yeah, so environmental justice is pretty heavily. Yeah, it does reflect. We don't have a strict program or subsection of what we do for it because it's basically interlaced in all of the programs we do. But obviously, working in the Anacostia Watershed, you can't do your work without things like that being a primary target and influence in all the projects that you do. So we work, as an example, we do a lot in stormwater education in a lot of the Hispanic and Latinx communities around Silver Spring, Montgomery County, some in Prince George's County. Trey, actually, our riverkeeper, runs Fishing and Friends program to talk about fishing and potential toxics in the Anacostia and stuff like that, with some groups around Prince George's County, Montgomery County. And then we also work on it pretty heavily in DC, especially with the long history of it along the main stem there.
Oh, great. Thank you.
Well, thank you all for coming out. Sorry, we're a little bit over. But again, thank you all for coming out. We really appreciate it. If you have any other questions, feel free to contact us, contact Pat, contact Ann. Our contact info is in there, I believe, and or on our websites. Hopefully we'll be collecting more data this summer, and be able to pass that along to y'all as well, every other week, just like we did this past summer and the summer before that. But with that, thank you all for coming out. This was a great presentation.